Hello, everyone. Welcome to the PGC Worldwide Lab. My name is Ole Andreassen. I'm chairing this session today, where the focus is the Nordic Tryggve Consortium, advancements in distributed computer system applications and beyond. And we have an excellent team of speakers today, starting with Louis from Karolinska, then Alex Frei from University of Oslo, Alfonso Buil from Denmark, and Kaarina Kowalec from Canada. And then we have two well-known people for the PGC, Stephan Ripke, who will be discussing together with Patrick Sullivan. And the overall purpose of this meeting is to show how we can leverage the Nordic advancements in big data registries, biobanks, and how novel tools targeted this opportunity can help us in discoveries in psychiatric genetics. So I think we can say that the Nordic system we developed is like a discovery machine. So then it's a pleasure for me to introduce the first speaker, Louis, Karolinska Institute, and she will give a quick update, the overview of what the Tryggve Consortium is. Please. Could you, we can't hear you, Louis.
Yeah, my apologies. I'm sorry. Hi, everyone. I'm Louis from Karolinska Institute in Sweden, where I focus on depression genetics and co-directs the KI Psychiatric Genomics Institute. I'm really honored to present our Tryggve network at this webinar, and I would like to thank PGC, especially Pat and Catherine, for the kind invitation. In this talk, I will give you a quick overview of our consortium and present the opportunities, challenges, and our progress. So Tryggve is an old Nordic, it's in old Nordic language, means safe and secure. It reflects our goal of conducting large cross-border collaboration on sensitive personal data. Pat, Ole, and Thomas started this idea way back and then established the consortium in 2017 within Sweden, Norway, and Denmark. Now it has been expanded to include all Nordic countries and Estonia. The idea is simple. We combine decades of registry information with recently invested genomic data to conduct registered genomic research in psychiatry. We are really grateful that a large number of Nordic researchers are now working with us in this consortium. It's a huge fun to work with such an outstanding team. Well, a large body of our knowledge in psychiatric epidemiology comes from the Nordic registers that have been collected over decades. For almost all residents in these countries, demographics and healthcare information were recorded across the lifespan and linked through this personal identity number. So in the Nordics, we really have a huge amount of patient-level data with rich information and very minimal selection. We know that Nordic countries are very similar in many aspects. For example, you probably heard of the Nordic model. That is, all these countries have a generous social welfare system. With that, you might wonder to what extent the data from Nordics are applicable elsewhere. Well, we looked into that. At least on the matrix of either socioeconomic status or morbidity mortality, the Nordic country cluster well with the other developed countries in Europe, Northern America, and Australia. These countries in the same color here. So the data from the Nordics are generalizable beyond this specific region. Now, with the recent investment of national biobanks and the large-scale genotyped cohorts, we now have 1.5 million genotyped samples with detailed healthcare records. Leveraging these register and genomic data, it means that we now have the opportunity to unravel the genetic causes beyond the case control phenotype. We can now look into clinical indicators such as severity, onset, or treatment-related phenotypes. However, just when we have all the data in place, regulations have tightened around the data privacy. Increasingly, individual-level data cannot be shared with other countries, so depositing data in a central server is not an option anymore. We have been thinking very hard about how we could efficiently contribute to the PGC analysis using harmonized analysis pipeline and phenotypes to really make sure that the node examples could align with other PGC cohorts, despite not directly sharing the data. With this in mind, we have developed the Tryggve infrastructure using the distributed containers. These containers have many advantages. The key thing is it allows the same analysis pipeline to run on local sensitive data with only the aggregated results shared externally. Now, please stay tuned for Alex's talk. In addition, we could harmonize the phenotypes from each country by comparing a wide range of epi and genetic epi measures. Here is one example that we did in major depressive disorder across Denmark, Sweden, and Norway. For example, here we compared the cumulative incidence of a specialist-treated depression and then derived the heritability from that. Despite the higher cumulative incidence in Sweden among both affected and unaffected for siblings, the pedigree heritability is actually comparable between these two countries. I am confident that at the end of this session, you will recognize the unique opportunities for psychiatric genetics research in the Nordics. We've made significant progress in infrastructure, which has addressed legal and technical challenges, and we are happy to share the tools and experiences with the whole PGC community. Many thanks to these wonderful colleagues in Tryggve, and thanks again for the opportunity to share with you here.
Thank you, Louis. It was a very nice overview. We have the Q&A session at the end, so that's why we move on directly to the next talk. And that's about Tryggve infrastructure from Alex Frei from the University of Oslo. Please, Alex.
Hi, everyone. So my name is Alex. I'm a member of Precision Psychiatry Group, University of Oslo, and I would like to present an overview of singularity containers and analytical workflows which we developed for Tryggve Consortium as part of Comorment Horizon 2020 project. So here's an outline of this talk, and I would like to explain what exactly are those singularity containers and which software packages we have included inside, then what analytical workflows we build on top of this software, and what are the typical steps that we apply in Tryggve Consortium for GWAS and PRS workflows. And finally, is our infrastructure really distributed or is it still a meta-analysis? Would be an important discussion point at the end. So starting with singularity containers, singularity container is actually just a single file that you include your software inside and to get the result of its dependencies. So if you have not used it before, here is how they work. You're probably familiar with the Plink software and how to use it from a command line terminal instead of if it is installed locally on your machine. So then you type Plink, that's a software name, followed by the arguments, which in this case is this -- help as an example. So when things are in a container, they are actually very simple. Really consistent. In the second example, we have a Plink software inside container called hello.sif, and we are executing it as shown here. Even better, you can actually define this variable called Plink in capital case and basically use your software exactly as before. So at this point, you'll probably argue that Plink is also a single file, and that's fair enough. So the real benefit is more obvious for a software that depends on specific operating systems, the libraries, or maybe Python or R, as for example is the case for LT-score regression analysis, where you would first need to install Python 2.7 and all of its dependencies.
Alex, your slides are not, there's a bit of
Oh, they are
some
not.
display issues.
Okay, 1 second. Let me see if.
There, now we can see it better. Thanks.
Okay. Apologies for this. So this is an example with LD score regression software where basically the workflow is exactly the same as before, even though it includes a lot more dependencies, both on operating system, Python, and modules. You bring a single file called ldsc.cif, and you use your software as before. So this consistency between how you install software once it is in a container is what I really like about this system. A few side notes. First, Singularity is also known as Obtainer, and that software cannot be containerized, so it has to be installed directly on your cluster environment, but this is now quite common. It's also possible to install it locally on your machine. Second, we use Dockerfile to make our containers, and this ensures that our containers are fully reproducible themselves and fully transparent about what went into those containers. And it also enables us to update the versions within containers pretty fast. As a third point, you still need to bring your reference data, and we discussed extensively whether we should include reference data within container or just put it next to it, and the second option seems to be more appropriate. So you still need to copy the reference data, but it's for the tools like for disc regression. And finally, one small caveat is about the hardware. So if, for example, the software itself requires GPU or any other specialized hardware, it will not work even if you bring it as a container. And for example, our containers are not compatible with today's M1, M2 Mac laptops, but almost everything else is compatible. So could you confirm that, that this is the next slide, that you can see it?
Yes, it is, but there's some layout issues.
Yeah, I'm afraid I'll just need to continue for the sake of time. I hope it's not too bad. So here is a selected list of statistical genetics software included in our containers, and they cover genome-wide association analysis, principal component analysis, polygenic risk scoring, and many other post GWAS tools. For instance, to illustrate the utility of this, I'd like to highlight metal. So if you use it from a container, it will be a latest version, which otherwise is not available for download as a precompiled binary. And about LD score regression, I already mentioned. So a very important point is if you use the software through our container, it's still very important to cite original software publication, not this overall container coached up bioarchive papers that we've put out so far. So please remember to cite original software. And beyond the software layer, we also have analytical workflows like GWAS or polygenic risk scoring, which I will explain in more detail. So these are the typical steps required when you apply GWAS and polygenic risk scoring inside our Tryggve Consortium. So assuming that you've installed this through the git clone, that's a specific command documented for installation of containers, and if you have sufficient resources on your high compute environment, then user would, at every site, would need to prepare the phenotype file and covariates, which we refer to as a master spreadsheet and data dictionary. Then prepare the genotype data, both QC and imputation and post-imputation QC. Then apply our GWAS workflow, export summary stats from the secure environment to send it to a central analytical team for meta-analysis, including Lee-1-out meta-analysis, and finally re-import back the Lee-1-out and perform polygenic risk workflow. So this is actually a meta-analysis, and I will get back to distributed part later in this presentation. So yeah, about this, we've talked briefly before. The point of this slide is just to illustrate that things are well documented.
Still, if you can hit it once more. Yeah, now it's better. Thanks.
Yeah. So things are well documented, and if you can try out if you try this out and documentation is not clear, please let us know. And as a specific starting point, I would like to suggest this hello.cif container, which is from hello world phrase. And then for this container, it's a very limited amount of files that you need to bring locally in order to give it a try. So it's a very small standalone package that does not depend on the other infrastructures that we developed. Okay, so in terms of high-performance computing resources, I would like to mention that it's only applicable to the workflows that I've described. The actual software within the containers themselves can easily run locally, either on your machine or on your secure server, and they don't depend on specific cluster environment like Slurm. But our workflows are currently dependent on a Slurm environment. So in the long term, however, we consider other workflow engines, such as, for example, Cromwell WDL, which is the main workflow engine, for example, in FinGen secure environment. So this is an important discussion, what kind of workflow engines such pipelines should support for more efficient GWAS and PRS analysis. So the next step for the users would be to prepare a phenotype and covariate file. We refer to this as a master spreadsheet because it should be just a single file containing all variables of interest, because later during the GWAS, a subset of the variables can be targeted as the phenotype of interest for performing the GWAS, and another subset can be targeted as covariates to include in the model. And if there are any other variables in the master spreadsheet, they will be ignored. It is also okay to have some participants with missing values or participants who do not have genotype data still to be included in the master spreadsheet, because the intersection will be handled as part of the GWAS workflow. This is just an illustration of some practical aspects that are included and taken care of as part of these workflows. This format specification is also applicable to polygenic risk score analysis. Of course, not for computing polygenic risk scores themselves, but then for evaluating how well polygenic risk scores explain the phenotype after controlling for covariates. And there are some nuances here as defined in a specification. For example, we expect also a dictionary file, like a data dictionary or code book, which defines at least the type of every variable. For example, which ones are continuous, which ones are binary and nominal. And for example, nominal covariates will be treated as factors and included as dummy binary multiple variables in the actual analysis. So I'll very briefly mention genotype and imputation. So most important for the users is to know that this pipeline.
You hit it once more, please?
I see.
Yes,
I see.
thanks.
That's an interesting side effect, sorry. So for the genotype data, we have Plink's, we support Plink's B file, the newest PGen format, VCF and BGen. But we do not include right now any workflow for performing imputation. And that's because most sites already have their own way of imputing. So sometimes by submitting to like a TopMed imputation server, sometimes locally with HRC panel or locally sequenced panel. So normally this is available, but it's a good discussion whether we need this as part of these containers. And what we can, in the long term, what we can include is the pipeline we've used in the imputation of the MOBA, Norwegian mother, father, and child cohort with almost 340,000 genotype participants with trios, mother, father, and child all included and genotyped. So this could be a nice addition to the workflows in the future. Then this is a specific script for performing the GWAS analysis. As you see, basically pointing to the right files, including phenotype file, genotype file, applying some QC thresholds, and then stating what would be the target phenotypes and what would be the covariates. And the good thing is that here the analyst can just choose, you see the analysis, dash analysis, Regini, and figures just specify that analysis should be done with Regini software and should produce figures also like QQplot and Manhattan plot. And this could be easily swapped with, let's say, Blink 2 or Sage. So there is this software itself will convert our input files with specification into what is required by Regini or Sage or Blink, and then convert the resulting summary stats into the common format specification for the summary statistics, as well as merge across chromosomes. So actually the previous step does not run the analysis itself, but just prepare all the input files and prepare all of the SLARM jobs, which can be then submitted after potentially reviewing those files if the user would like to double check that things will be done as expected. And the output, as mentioned, is a harmonized format regardless of the GWAS software, merged and with some brief report made at the end. And that's of course a standard GWAS output, QQ plots height for height, and just an example. For the meta-analysis.
It once more.
Yeah. Yes. Thank you, Ole. So for the meta-analysis, we do not have a specific workflow, just because it's quite simple and it's performed centrally anyway. So, but it's right now we are doing standard metal, and the other software GWAMA is also available, as well as rare GWAMA. Then final step about the polygenic risk scoring analysis is quite simple, and most of the flags here are quite optional, because quite optional if the goal is just to compute polygenic risk score. They are only required if we should also evaluate how well PRS performed in terms of Nagle-Hirsch R-square and different beans of the odds ratio between different beans of the polygenic risk score. So that's a brief overview of the pipeline. There are things well established like GWAS or polygenic risk scoring, and then some modules think we need to work on them more, or even introduce from scratch like impute model. But we have already done quite a bit of work to include into this. So I think probably one of the most important slides is this. It's a discussion about meta-analysis versus distributed analysis. And so far, as you see, we focus on meta-analysis, basically streamlining analytical workflow at each site. But because this worked so far very, very well in a GWAS setting, but in the future, I do expect there will be applications which require full distributed analysis, which involves an iterative procedure, something like an EM, expectation maximization, like algorithms for jointly fitting a single model across multiple sites. And these are just a few ideas of concrete examples where this kind of already might be important. We just probably don't do it. For instance, in the mixed effect models for GWAS like Regini, we do improve power by this local leave one chromosome out scheme, which potentially could further boost the GWAS yield if those polygenic scores controlled for are based on leave one out summary stats and not just locally. And that's also applied in a more general setting. Like we are now working on a tool called FEMA GWAS, fast and efficient mixed effect model for longitudinal GWAS. And there we also do expect that there will be an iterative procedure about fitting fixed and random effects, and fitting random effects should be done on a meta-analysis. And then also basically, if we move beyond the marginal association in the GWAS to joint association with multiple regression across multiple SNPs, as we do in our polygenic hazard score in some survival analysis frameworks, this may also trigger the same question: can we jointly analyze all of this data rather than rely on this manual workflow about leave one out, bring in each leave one out, then back to every site, etc., which kind of is already distributed with 2 iterations, but it's not fully automated. And personally, I think it's basically an easy task to implement the software, right? Not easy, it's a meta development, but it's solvable. And we can expand the containers with such distributed models. It's a medium hard task to align the phenotypes and data dictionaries across sites, and we really progressed quite far on this within Tryggve Consortium. But the hard task would be to open up IT systems and bring all of our software and then the systems wrapping this software into like endpoints that can talk to each other over a network, like pass this all through security reviews, because we are lucky to be within those secure environments where for most cases we can just bring our data as long as we have rec approval and work within those environments. But opening them up in a secure way, that to me sounds like a very hard task. And we are working on this. We have our advanced user support with IT people, at least in University of Oslo, but it's interesting kind of how to implement it across several countries. That basically concludes my presentation. As a summary, we've developed
one more hit.
Thank you. So we've included the underlying software and built analytical workflows around it. And this, to some extent, hides the complexity involved in running GWAS and the PGS analysis. it's a very important discussion how this overlaps with the Ricopili pipeline and what should we learn from it to improve this further, as well as we've discussed briefly limitations of current meta-analysis. And the most urgent example would be to detecting sample overlap across different sites. We have some solution for, but it's not, I think, needs to be developed further. And with that, thanks a lot for the Comorment project and for everyone participating in this, both development, designing and testing the containers. And for further information, there is a preprint and the code is publicly available with all containers shared on this GitHub repository shown below. Thank you so much.
Thank you, Alex. It's a very clear overview of the technicalities of the infrastructure, analytical infrastructure. And I think this is a good example of how the infrastructure is fitted or the methods are specified to make the most out of the Nordic data. But then we will move on to see how a couple of examples, how this infrastructure has made it possible to do some new discoveries, like Nordic efforts in major depression and schizophrenia. And first, there will be Alfonso Buil from Copenhagen, Denmark. Please.
Hi, thank you. Can you hear me? Can you hear me well?
Yes, yes.
Okay, perfect. So yes, I'm Alfonso from the Institute of Biological Psychiatry in Denmark, and I'm gonna give an example of the use of these containers with putting together information for the Nordic registers and the big genotyping platforms for 5 different countries. The goal is going to be to compare the genetic architecture of early onset and late onset depression. This is the main team that has done all the analysis that I'm going to show in the next slide. So I was saying, main aim is investigate differences in genetic architecture between early and late onset depression. And this figure summarizes everything we've done so far in this project. We have different data sources from these 5 countries, Nordic countries, and we have defined the different phenotypes. We define early onset depression as having the first diagnosis earlier than 25 years old, late onset depression having the first diagnosis after 50, and then broad depression is any depression that you have regardless of age. And yeah, for early onset depression, we could put together individual cases for 5 different countries, and there was more than 45,000 cases, more than 100,000 controls. For late onset depression, we get a bit less because we didn't get cases in Denmark and Norway, so we got 37,000 only, and in total we got like 150,000 cases of broad depression. So we did GWAS for all these phenotypes, but also we have in progress a case GWAS, just comparing late versus onset. We don't have results yet. So we are doing GWAS for all of these phenotypes, and then we do some functional annotation to understand the biology underlying these signals. And then we perform a set of analyses, all of them trying to compare, contrast this early and late onset MDD. We do a lot of different ways of looking at the genetic overlap between these phenotypes and other psychiatric and somatic disorders. We compare SNP heritability. We also look for unique overlap using genomic STEM. We did some Mendelian randomization to explore causation, and also we did some analysis on prediction. I'm going to go very quickly through the results because we don't have much time. This is the GWAS for broad depression, and then it's the early and late depression here. We have some hits, but not that many for early, and only 2, I think it's 8 for early and 2 for late onset. We compare heritability, the SNP-based heritability, and we see that our estimate for the early onset depression is around twice the size of the late onset depression. So this is something that has been published before, that early onset depression has a larger heritability than late onset depression. When we look at the significant loci, I mean, we see that the ones that are significant in early onset depression are not, in many of them, they are not even nominally significant in the late onset depression, but still we are not, I mean, we know that the late onset depression is a bit less powerful, and we are still waiting for the case analysis to talk about loci that are specific for early onset depression. Regarding the functional annotation, we saw something kind of interesting, and that is that the only tissues where any of these phenotypes were significant was fetal brain. So we didn't observe anything in the adult brain, but most of the fetal brain annotations were significant, but only for early depression, early onset depression, and not for late onset depression. Looking at the genetic overlap between these definitions of depression, early and broad, well, they are quite overlap. Genetic correlation of 80%, late and broad a bit less, and early and late around 60%. So the overlap is not complete, quite far from complete. And then when we look at genetic correlations with other mental disorders and with other somatic conditions, we see that for early onset and late onset, they go always in the same direction. In most of the cases, the estimates are similar, but there are two cases in which they are very different. One is suicide attempt, that the estimate for early onset depression is like double to the late, and the same with autism. In this case, the difference is even larger. It's like 3 times. So the genetic correlation of early onset depression with autism and with suicide risk behavior is different and larger than with late onset depression. When we try with genomic SEM and Mendelian randomization, I mean, genomic SEM trying to control, to look at the overlap of early onset depression with some conditions, controlling for late onset depression and the other way around, we see that early onset depression has a unique effect for suicide attempt, again, and for some of the cardiovascular conditions, and we don't see that for the late onset depression. With Mendelian randomization, we didn't see striking results so far. Most of the things are not significant or very close to zero in any case. Finally, regarding prediction, we saw that the early onset depression PRS predicts better than the late onset depression PRS for any condition in depression, but also for other comorbid psychiatric disorders. And in fact, the early onset depression, even that it's a much smaller, it predicts almost as well as the broader depression. And finally, this is a very preliminary result. We are also looking at the how we can predict suicide risk using the early onset depression PRS, and we see clear differences between the top 20% and the bottom 20% in Estonian data so far. This is quite preliminary. So as a conclusion, we see these differences between early onset and late onset genetic architecture, with early onset having higher heritability, enrichment for fetal brain function, larger overlap for suicide attempts and autism, and better prediction for suicide behavior risk so far. Thanks, everybody. Great.
Thanks a lot, Alfonso. Great example of how the unique features of the Nordic registers combined with genotypes can be utilized. So then we move on to the next example. That's from our transatlantic Nordic collaborator, Kaarina Kowalec from Manitoba in Canada. Next example is schizophrenia, please.
Perfect, thank you so much. So hi everybody, I'm Kaarina Kowalec, an assistant professor at the University of Manitoba. As Ole said, in Canada, sort of, I guess, honorary, maybe Nordic member. We are in the Nordic part of the world, I suppose, but certainly don't speak the same languages. But yes, I'm here today to talk about a GWAS that we're performing on schizophrenia, specifically in Sweden, so hopefully to expand beyond one single country in the near future. Okay, so as everyone is probably very well familiar with the PGC efforts towards understanding the common variant map of schizophrenia in the past many years, there's been huge progress in the last 15 or so years, I guess I would say, in terms of understanding and identifying more really reliable loci within schizophrenia cases and controls. And so now we're at to just under, I guess, about 300 loci that have been really well replicated. And so from there, what we wanted to talk about really was S3. So many people probably have heard or kind of the name S3 over the years, but the Sweden Schizophrenia Study, what S3 stands for, of course, located within Sweden or started in Sweden. So what it is, it's a case control study of schizophrenia that from people that are from Sweden. So cases that were identified using the register data, as Lee had pointed out, using hospital register data, inpatient and outpatient admissions. And so in terms of the historical context, S3 has played a really large role in the PGC schizophrenia GWASs. And so where do we take this resource now? So we've collected a lot of different samples from many different studies and pulled them all together. And so where do we take this now that we're at a really sizable number of samples? And really, how do we go beyond the traditional sort of minimal phenotyping instead of just case control status and moving towards understanding or incorporating the information from someone's sort of longitudinal entire kind of lifespan to better understand genetics of this disorder? And then lastly, also, how do we collect further samples? And so more efficient means of collecting samples. We know that nurse-led collection or getting people to come into clinic is very challenging and expensive, and we don't get everyone. Of course, we only get the people that volunteer or are able to get to the clinics in order to get samples from them. And so our objective overall then is to understand, of course, the functional significance of schizophrenia genetic variation and further understand the genetic landscape of this disorder. And so like I was saying, in 2009, it was really one of the first publications where the S3 samples were contributed to sort of a PGC-like effort with about 500 cases and around similar number of controls, leading up to where we are now with at least more than 50% of these have not been reported. So we have about just under 11,000 cases schizophrenia and 14,000 controls, so sizable increase. And so again, so how do we go beyond this sort of traditional minimal phenotyping? So looking at a snapshot of someone, so when at a single time point, so a cross-sectional kind of assessment and move to a womb to tomb. So we have this type of information, and how can we best integrate it into understanding genetics? So like Lee was mentioning earlier, so in Sweden, just like the other Nordic countries, there's really extensive register data available to conduct these sort of registered genomic studies, anything from causes of death to multi-generation, meaning sort of family pedigrees, military conscription records, which is sort of like IQ measurements, and then of course the more commonly used inpatient, outpatient records for hospital information, and then drug information for prescribed medications. And then secondly, sort of how do we go beyond sort of the traditional collection of samples? So like as has been done more recently by IPSych in Denmark. So when a baby's born, they get their heels pricked for some metabolic screening and put into sort of what's traditionally called like a PKU biobank or a dried blood spot biobank. For research purposes, we can get a sample from these dried blood cards. They're typically stored for anywhere from 18 to 25 years, typically in some sort of refrigerated manner, and then we can use those to get DNA. And like I was saying, IPSych has been doing this with that sort of the forms the basis, I guess, of what IPSych was really about, and has published extensively using these samples for GWASs and many other purposes. And so now we're trying to do this very similar in a similar manner in Sweden. And so it's available on virtually all individuals born in Sweden after 1975. It's representative of the entire population. So of course, not only those coming to a clinic or volunteering for research, representing really one of the biggest, I guess, or one of the largest benefits of using these samples, because of course it's not, it's more of a population base as opposed to a more biased research collected manner. And so our analytical goals are combine our samples then with, of course, perform meta-analyses with the latest PGC papers. So I would be comprising of just the European samples anyways of 42,000 cases, and then also eventually with the other answers as well. And then next thing would be to check in, of course, with the SNP heritability, genetic correlations, as per the sort of standard GWASs would perform with other disorders and traits. And then we can also compare it with different phenotypes for schizophrenia. So we can use we have sort of a broad and a narrow definition of schizophrenia based on the number of times that someone has come into clinic. And we can also incorporate other pieces of information, such as antipsychotic use and things like that. And then performing longitudinal GWAS modeling. So instead of, like I said, using the case control status as sort of the outcome, using other forms. So this is using more longitudinal information. So most of these use like age of onset or family history to account for those types of aspects. And then also other outcomes like maybe number of hospitalizations over someone's lifetime. And of course, performing functional annotation and looking at how these compare to even some of the rare variant findings. So in terms of the cases that have been genotyped so far, I won't focus on the controls here, but you can imagine the number is just a slightly higher number for the controls. But as you can see, the blue represents sort of the past collection that everyone would have seen in some of the PGC studies. And so on the left-hand side of the plot, you can see, or the pie chart, you can see those that were collected in the last few years. You know, half of those coming from the PKU or the dry blood spots, and then some of them from other measures of collection. And so big thanks to Robert Carlson for pulling all this data together in terms of pulling the genotypes all together. And so most of them were on Illumina arrays. And so this is representing 17 batches from 11 different waves or studies. So an immense amount of work performed by Robert. And then they've all undergone the standard root copy QC, and of course, agency imputation. And then in terms of looking quickly just at some of the heritability. So for example, so S3 has a higher heritability than in comparison to, say, the most recent schizophrenia 2022, if I excluded the Swedish samples. But of course, the end goal would be to meta-analyze them all together, at which point you expect probably the heritability to be kind of somewhere in between. When comparing then the genetic correlation then with a bunch of different traits, and again, this is sort of preliminary data. We still have one more cohort to add into our sample set, but you can see that, for example, the bottom two represent the narrow and broad definitions. They don't vary very much, so sort of kind of giving us the inclination possibly that we could use more of a broader definition, but more to be seen. But it aligns with a lot of the PGC or with the most recent PGC schizophrenia GWAS, which is nice to see. And so quickly in conclusion, so we can move on to some of the discussion points. So really this carefully phenotyped cohort can make a really meaningful contribution to understanding the genetic architecture of schizophrenia, and certainly more to be seen. And then, like I said, expanding to the other Nordic countries, I think will be really important to see if we can find any additional genetic information or further understand the genetic architecture. So thank you so much.
Thank you, Kaarina. Looks like you're going to be really exciting new findings there. So, but in order to move on and to the discussion, we will now have the chair, the discussant, Stephan Ripke, who will, together with Patrick Sullivan, chair the discussion. But I just need to remind everyone that, you know, in the beginning, like 10 years ago, we started a lot of this work, and then we had a cabin. We rented a cabin in the mountains of Norway. And I think while Stephan was there out snowboarding, Dr. Sullivan, he was really chairing the scientific sessions. So they all contributed really in the beginning there with the Nordic approaches. So Stephan, can you facilitate discussion?
Just 1 second. I'm going to ask Alimar to turn the Q&A on so we can start getting questions in the chat or in the question and answer thing for the panelists to handle in a bit. Sorry.
Thanks.
Okay, I'll do.
Sure. So I mean, I can start this off a little bit since I'm the official discussant here. So first of all, congratulations. I'm not an active member. I was just in the beginning. I was a little bit there again in Norway, but like I'm not an active contributor, and many of these things here I saw for the first time, and I'm super happy to see this progress. I'm very impressed. Obviously, a discussant is here to summarize a little bit what we have seen here, but I also take my role here also to be a little bit, let me be a little bit also provocative or asking questions so that we can get maybe also the other people here that are on the call to have some questions here. But don't get me wrong. First of all, I'm really impressed and I love it what you're doing here. Let me share some screen and just give, maybe while we're waiting for some questions, give me some 5 minutes to summarize a little bit what we have seen here, what I think is the most important outcome here. Obviously, these are still from Yuli, these beginning slides, and I understand it and take me wrong if I'm wrong, but like I understand that. Sorry, my phone was just going on. That this consortium, Tryggve, or at least this webinar that we have here today is actually trying, or it's very successfully doing so, setting up the stage for two a little bit different topics here. The one topic is really to present this collaboration of these Nordic countries that we see here in this first slot, and I will come back to this in a second. The other thing is to set the stage for computational structure in order to enable us doing analyses across centers without sharing genotypes, which will be a big task for the future. But I think we can keep these two tasks a little bit separate. They're just a little bit coincidentally in this wonderful consortium here right now. So let's go first a couple of slides. I think this is, for everybody, this is probably one of the most important slides that we see here. These countries that are coming here together with having so many millions of people in this absolutely great resource of really all this clinical data basically being very centralized. And this is very different to many other, especially European countries, and from Germany, where I'm from. The US also doesn't have that so centralized, and also so this is absolutely fabulous. And also I understood this question. I just wanted to make the short point here. You have very generalizable numbers for morbidity, etc., and so on. The one thing that you're not, and this is what the Nordic countries have way better than the other countries, is what is not generalizable is exactly this centralized phenotypes that I was just mentioning. So this is the fabulous thing that you have here. The one thing that I again want to be a little bit provocative. I want to make a little bit the question mark to now functional, and I just want to make this a little bit here. So because let's talk about this. I want to put these questions out here because there's this other slide that actually Alex showed a little bit later, is that there's a couple of things still in yellow. And don't get me wrong, I don't want to be super critical and say like, oh, you didn't do your job. But like, as long as there's almost half of that yellow, I question a little bit the statement we are functional and this works here right now. And I will get to this a little bit in more detail here right now and ask the questions that I didn't find in these slides yet. And this maybe starts the discussion here. I don't want an answer right now, just asking the questions here. For example, this wonderful presentation that we had here from Alfonso, this early onset and late onset, for example, where you have here 5 different countries with 46,000 cases. I want to ask the question first, and because I didn't see this on these slides, have any of these containers that were presented before, have they been used for this analysis, or is this an analysis where you classically just share summary statistics and somebody is doing the analysis centrally? So I think that's an important question to ask you, because like, if my assumption is right, that this is still an old-fashioned summary statistic based, then I would just, again, make a little bit this question mark, this is already functional or is already used. The other thing that I want to just pose a couple of other questions here, this very nice plot. So you very nicely, and I don't question this at all, you very nicely make the point that early onset MDD seems to have stronger signal here. There's no question about this, and this is wonderfully shown here. A little bit more detail about hits that are unique to early onset in the long run. We probably want to look into this before we have a definite statement, oh, here we see a very big difference in here. This can be sample size driven. It can be just a random effect that just passing barely the genome-wide significant hits. So this needs a little bit more follow-up, obviously, in here. But again, you showed very nicely this overall signal in here. Coming to this other presentation about schizophrenia in here, and I think there's the most important criticism that I have here. I mean, obviously, wonderfully presented, no question. And absolutely impressive. These 10,000 cases that we have here right now, absolutely fabulous. Obviously, the criticism is here. We wanted to have a webinar here about Nordic countries and especially this infrastructure to do trans-country analyses, and then a presentation about Sweden is obviously not exactly what we want to show here, even though, again, congratulations, these heritability estimates and these coheritabilities are wonderful, and I'm very much looking forward how this will look like, though, again, a couple of things are a little bit, the meta-analysis with PGC obviously is something that I would love to see. So again, this presentation was clear, very good, and highly thrilling, but it leaves a couple of questions open that we want to see at least in the near future. And I think that's where I made most of my points already now. And forgive me if, again, I don't want to be too critical, right? So it's just a couple of discussion points in here that I want to put out here, and maybe then we can open up already some people, or maybe had wants
maybe
to.
I can just, like in 30 seconds,
yes.
to answer you first this question, because this analysis in the early versus late onset depression, as you said in the beginning, with the whole project, has the two sides. We have, of course, the scientific interest, but also has been conceived as a proof for the containers. And then the analysis, what is the GWASs and the PRSs, have been done in all sites using the containers. That was part of the thing. We really wanted to try it.
Yeah.
And we took that interest that we had, scientific interest, as an excuse to try the containers, even that in some cases it took a bit more time. But
Okay, wonderful.
And
so
regarding the other point, you are. I mean, it's true that we still are. Some of the analyses are still in progress, and we have to finish this case and look more carefully at these logs. I mean, there are things that we still have to iron for the final analysis. But yeah.
Yeah. Okay, I understand. And so it makes me happy to hear that you're using these containers and that they're functional. And there was one point that I really wanted to put out here. Also again, very provocative, a little bit. And Alex, don't get me wrong, you did wonderful job setting all this up. I want to put this one. This one could be. I mean, I'm invited here, so that's obviously then the people have to live that I obviously go a little bit harder on these points here. So you mentioned that QC phasing and imputation is not integrated yet because most people do this already on their side. I agree that this is probably true for especially these big biobank things where they really have all their power in there, be it UK Biobank, etc. I disagree a little bit. I hope I don't know, but like especially for the schizophrenia collection that we saw here right now that was presented, for the NDD collection, probably these are GWAS sets that the people here did their QC and imputation themselves, and especially then, again, I'm the person who has done like so many QC and imputation, and I think this is absolutely crucial step. This is not a step that we should keep just for everybody to do themselves.
That's absolutely important. I fully agree. So kind of the trade-off on our end was, we cannot share and provide a reference panel. So the best reference panel out there is TopMed. So we would still need to ask people to use TopMed is possible, right?
Yes.
And then if not possible, they will do everything locally, probably with an HRC panel, which we also cannot share, right? But at least we can facilitate scripting and workflow if they have access, if they established access. So kind of having those things on the back of our mind, we decided not to say that there is kind of from our side, there is an absolute universal way of doing this. And then in the Nordic collaboration, every site already had kind of imputation performed, right? So we decided to go with what we have. But it's a very crucial point, and I'm totally open to bringing what we've done, let's say, with MOBA, Norwegian Mother, Father, and Child cohort QC and imputation as kind of standardized workflow to this. The question is how much value it would bring if that still brings us back to the less than state of the art in terms of the reference panel. After 1 million reference genome Europe, that's probably would be different.
Yeah, I mean, just to make two short additional comments. Like, so first of all, QC does not need a reference panel, right? So this could be provided for everybody to do it in a standardized manner, and everybody is doing it differently. That's why I'm a little bit. So that's the one point, and the other point is like, I understand, and I fully agree. TopMed is definitely the strongest one here, but like Tryggve has this thing that you cannot share genotypes, right? So just maybe somebody can comment on that, but if you cannot share genotypes, you cannot use TopMed as well, or am I wrong here? Yeah, Pat wants to.
If I can take that, yeah, we looked into this extensively. So it's possible to actually make your own private imputation server. The software that's behind Sanger and TopMed and Michigan Imputation Server is actually open source. And so you can actually clone it and put in your own data. This has been something we've been trying to do, but getting a framework for that has been tricky. This is also something the PGC is facing because the HRC, the Sanger site, has not been updated for ages. The data sets are clearly have been surpassed by other ones. And actually getting permission to send European data to TopMed is really difficult. It's actually impossible from our perspective. So this is an unsolved problem that we've been trying to think about. We've sort of kicked it down the road a little bit because getting this project to that position was the hard step, but the imputation is something as a conversation we need to resurrect for sure.
Yeah. I fully agree.
So if you can't chip in there, remember, Pat, we spent so much time, you really pushed this, and it ended up there was some political and really, legal issues we ended up with. So, but that is being solved as we go now, and it's really, really an important topic.
Yeah, essentially we need a private, we need a Tryggve, a safe, secure imputation reference, an imputation server for this part of the process, because it's absolutely critical. And we tried to actually get a couple of the key data sets from TopMed, and they wouldn't let us get them if we were going to establish an imputation server, because we figured, if we get the 3 or 4 biggest studies from TopMed, that would allow us to do it, but they said no.
But could I, moving forward, bringing the Nordics to the next level, maybe a quick question from me to Stephan and you, Pat, that, this going beyond like just a quick standard way doing like meta, this iterative process and going back and doing polynomial scoring, leave one out and stuff, the next level. I think that, what do you think about that? I think you, Alex, had this as a kind of a discussion point. I think that is super important for us to strategize where to go, where to put our resources.
Absolutely, and it's nice to hear Stephan urge us ahead in those couple of regards, because yes, this is a critical part of the whole thing for all sorts of reasons. And we take his comment, I think, in the spirit with which it was presented. So thank you.
Are there any
Q&A now?
please.
Anybody else who has some questions? Is this open to everybody, to all participants? Because it says here 47 participants, but we cannot see any names, right? Ayemera, do you know?
Yeah, there's no questions currently.
Okay. I mean, Pat needs to leave in 2 minutes. And my weekend also began an hour ago.
But maybe on Stephan, for you, you've been doing the PGC for so many years now, all kind of focused on the central, but how do you see this more like not only in the Nordics, but there must be several samples also in your kind of history that were not shareable? Quick answer.
Quick answer is I don't have a good solution to that. The only, the most important solution that I have is that I think like the most modern imputation references are way less important than getting all the data together. This is very clear. I mean, collecting huge sample sizes is the key trick. It's not the most modern imputation server. And a very good trick is to have everything on the same imputation. So that's why I do not mind. I would even do like HapMap imputation in the end, right? If all samples are coming together. But nowadays it looks different. The people cannot share data anymore, and we have to deal with summary statistics, and we have to deal with possible sample overlap and all these things. I don't have a full answer and good answer to that yet.
Thank you. I think it's my job as a chair to thank all the speakers and the discussants. This was a great session. Thank you all. And we even had a very good feedback from some of the audience. And I thank you all again. I think there's a lot of opportunities in this, both the Nordic region and in this container technology, as Stephan said. And thank you all for making this into such a great session. Thank you, and have a nice weekend.
Thank you.
Thanks, everyone.
Everybody.